Okay, hello everyone and welcome to the February meeting of the Global Health Youth Organization. My name is Noraldine and I'm a co-founder of the organization, and we are very excited to see everyone who joined us today. We want to thank everyone for joining the meeting and working around any scheduling or time zone issues that you might have had. As for the format of today's meeting, we will begin with a quick introduction of our speaker, followed by his presentation and then a question and answer session. After the Q&A section, we will be posting the shadowing certificate link in the chat, and we ask that all members stay muted throughout the presentation so that we can limit interruptions and distractions. Please feel free to add any questions or comments that may arise in the Zoom chat. I will now be passing it over to Adam, the other co-founder of the organization, to introduce today's speaker.
Thanks, Nor. So hello, everyone. And as Nor mentioned, I'm a co-founder of the organization. It is so great to see all of you that joined or are happy you're able to attend this meeting. We want to once again thank Researcher Sairaj Sajjath for taking the time to present today. To quickly provide an introduction, Raj Sajjath is a researcher in a cell biology lab at the Rockefeller University, as well as a PhD candidate. Raj Sajjath is heavily involved with research on the role of tissue stem cells in inflammatory skin disorders. Please remember to stay muted throughout the presentation to minimize distractions. Again, if any questions do arise, please type them in the chat and we'll get to answering them at the end of the presentation. With that being said, we will now hand the floor over to Raj Sajjath for his presentation. Thank you.
Thank you. Oops, that is the end. All right. Yeah, thank you, Adam, for that introduction. And I hope you guys are all having a really good weekend. And I hope anyone that's in the US is bearing this this massive cold front that we're having. And I put my jacket on because my room is kind of freezing. So I hope that's not too distracting. But either way, I hope you guys are all having a great weekend. And like Adam said, my name is Raj, and I'm a second-year PhD candidate at the Rockefeller University studying the biology of skin stem cells. And what I want to talk to you guys about today is a set of global health challenges that are very closely related to the work that I do, namely inflammatory skin disorders. And before I start, I just want to do some acknowledgements. So first, I want to acknowledge my lab. We're a big group of almost 40 people, so I'm not going to show everyone, but I do want to thank the quote-unquote inflammation team, which started with Dr. Shruti Naik, who's now a professor at NYU, and Sam Larson, who's now a postdoc at NYU, and has since continued with Chris Cowley, who's a senior grad student at Rockefeller, and most recently me when I joined in May 2020. I also just want to thank our university, as well as the Howard Hughes Medical Institute, which really gives our lab a lot of really helpful external resources and funding. And of course, I want to thank the Global Health Youth Organization for putting together this really wonderful series and for inviting me to be a part of it. I think one of the very few nice things about this pandemic is that it's really made research and science more accessible around the world. And I think organizations like this are really one of the main driving forces behind that increase in accessibility. So I really, really praise the organizers for setting this up and running these talks. And I really thank the audience for attending as well. Okay, so to go over my overall presentation goal, I'm a basic scientist. And what I'd like to do here is to illustrate some of the roles that basic science can play when it comes to tackling global health challenges. So here I've laid out a very reductionist outline as to how we tackle a number of global health challenges, starting with basic discoveries about the nature of whatever biological agent is causing the problem, then going into more translational research that starts to directly investigate ways to tackle the problem, and then clinical research, where promising solutions are tested within relevant populations. And lastly, what I've just termed practice, which broadly refers to like distribution, policy, economics, et cetera, that's needed to actually get the solution out into the world. And one important side point that I want to make is that this view of responses to global health is useful for global health challenges is useful for my presentation, which we'll see in a bit. But I really want to emphasize that this is a very therapeutically oriented view and definitely doesn't cover the full spectrum of an adequate response to any global health challenge. And I've added a note here that treatments are important, but aren't the only thing that matters. And I think just as a quick kind of soapbox side point, I think our current situation can really help us appreciate that point, where we've seen that understanding COVID to the extent that we can develop, test, and distribute a vaccine is fantastic, but that there's also a whole slew of other political, socioeconomic, educational, racial, et cetera problems that are really essential to understand and address as part of the whole response to any given challenge. And I really think that anyone working within any challenge or global health challenge should be at least tangentially aware of these other issues. So this said, I'm a basic biologist studying both health and disease. So what I want to show is how one part of the response to a global health challenge, specifically understanding the underlying basic biology, can contribute to one part of the solution, namely effective treatments against whatever is causing the challenge. So it's in this light that I'll talk about some of my lab's work related to inflammatory skin disorders, which comprise some of the most prevalent and burdensome diseases worldwide, and specifically show how asking a basic biological question of how does the skin respond to environmental stress has led to important and novel insights into this currently uncurable set of diseases. So before we jump in too deep, I want to just quickly first show you guys why we as basic biologists think the skin is so fascinating and important to look at. So just doing some quick anatomy. If we zoom in on a piece of skin and take a cross section, we'll see two basic components. The first is the epidermis, shown on top, which makes up the quote unquote skin part of our skin. And in normal conditions is constantly being rejuvenated by a pool of stem cells at the bottom of the epidermis, which when they divide um, can either form new versions of themselves, shown by the circly bottom arrow, or differentiate upward to form the various layers of the skin, which eventually get shed off as dead skin. And the second component is an underlying layer called the dermis, which comprises a number of supportive cell types like nerves, blood and lymph vessels, fat, immune cells, and etc. So this established, a lot of people know that the skin or the collection of these components is the largest organ in our bodies. But one thing that's often less appreciated is that the skin is also one of our primary environmental interfaces and is constantly exposed to a ton of dangers like pathogens such as viruses and bacteria, wounds, and noxious or poisonous agents. And in order to be able to respond to these stresses, or this really diverse set of stresses, we've evolved really elaborate networks between our skin cells and these supportive networks to help our skin maintain itself in normalcy and repair itself upon stress. However, one really important point to note is that these processes need to be really tightly regulated or controlled, since we also know that a number of diseases occur when our normally restorative repair processes go out of control. So here I'm showing 3 hallmark-related disorders, specifically psoriasis, atopic dermatitis, and chronic wounding, which I'll spend most of my focus on in this presentation, but just be aware that there are many other skin disorders that also arise from dysregulation of otherwise normal repair processes. However, the main takeaway I really want to give from this slide is to impress on you guys that the skin constantly needs to respond to environmental stresses, and that any misstep in normal responses to these stresses can lead to disease. So these concepts are really the motivation behind us asking our earlier big picture question of how does the skin respond to environmental assaults? And in this regard, one thing we know pretty well through really decades of research is that dealing with almost any stress requires our skin to mount an inflammatory response, which comprises a set of immune responses that commonly drive this bottom layer of stem cells to proliferate or expand within the damaged skin and then orchestrate tissue repair. So the inflammatory responses present in skin are generally well characterized, meaning that we know a good amount about what cell types and molecules, et cetera, are activated in response to a number of specific environmental assaults. However, a lot less is known about the enduring consequences of these inflammatory responses in skin and how these consequences bear themselves out in both health and disease. So this is the primary question that our group is interested in right now with regards to inflammation. And we really think that this has a lot of potential to teach us first at a basic biological level about some of the principles by which our tissues respond to bodily or physiological stresses. And second, at a disease level, we really think that asking specifically about the long-term consequences of inflammation can help us understand how a number of prominent chronic disorders work. And since this is a global health related talk, I want to spend a little bit of time on the second part, or the disease side, and show you guys that the diseases that we're talking about, which are related to our research, really are a major global health challenge. And again, I'll focus on 3 of the most prominent disorders, specifically psoriasis, atopic dermatitis, and chronic wounding. So inflammatory skin disorders can broadly be separated into 2 groups, the first of which is diseases with recurrent pathology, and the second of which is diseases with persistent pathology. So recurrent pathology means that inflammation will often flare up, go away, and then come back later, often in the same areas of skin and often with greater severity. So the two hallmark cases of recurrent disorders are atopic dermatitis and psoriasis, each of which affects hundreds of millions across the globe every year. And although the true economic burden of these diseases is difficult to estimate, it's safe to say that each places an annual burden of tens to hundreds of billions of dollars across the world every year. And similar to these diseases are persistent disorders, which are another form of inflammatory skin disease. But instead of inflammation going away and then coming back again, inflammation just never goes away and gets worse over time. So the hallmark persistent inflammatory disease in skin is chronic wounds, which, just like they sound, are wounds that just never heal. And although global statistics are hard to come by, I think we can appreciate just from the US statistics that this disease affects a huge number of people, tens of millions in the US alone, and is associated with a very hefty economic burden, and in the US alone estimated around $25 billion a year. So we can really see that these diseases make up collectively a major global health problem. And while we have measures to reduce certain symptoms of each disorder, we still don't have a cure for any of them. And we really think this boils down to our lack of understanding at a basic level of how inflammation is actually dysregulated in these chronic disorders. In other words, we don't know why inflammation keeps coming back in the same places and often with greater severity. And one really powerful way we can begin to tease these questions apart and really work toward a cure is by returning to our basic question of what the long-term consequences of inflammation are in skin. And like I mentioned, this is really one key area of investigation within our lab and a question that I'm personally working on to address. However, I'm still a new graduate student. And while I love to talk to you guys about some of my work or about some of my own work, I don't want to show you guys at this stage too much preliminary or unpublished data. So in this regards, one thing I really do want to show you guys is the work that really jump-started this line of investigation in our lab. So this is carried out by Shruti Naik, then a postdoc in our lab, and again, now a professor at NYU, and Sam Larson, who was then a grad student in the lab, and now a postdoc at NYU. And almost all the data I'm going to show you guys is from this paper shown here, which is published in Nature in 2017. And going forward, I'll expect a really basic kind of intro high school biology background from you guys. But even if you don't have that, I'll try to make things easy to follow, and I expect that you'll still be able to get, the really big picture points that I'm trying to get across. Okay. So to really ask this big picture question of what the long-term consequences of inflammation are in skin, we first need to settle on a way to model inflammation in an experimental setting. And to do this, Sam and Shruti used a drug called imiquimod, which induces a psoriasis-like inflammation when applied to the ear or back skin in mice, which are our main animal of study. And they followed this general experimental structure shown on the bottom, where they inflamed mice for 6 days with imiquimod, which is how long it takes to get to the peak of inflammation with this drug. And then they did nothing for another 24 days through day 30, by which point the mice resolved inflammation and were in what we call a post-inflamed state. So this point on post-inflammation or resolution is important, so I'll really just briefly hammer on it here. So on the left is the same experimental schematic that you guys saw earlier, and on the right are cross sections of skin at the peak of inflammation at day 6, shown for untreated and imiquimod treated in the left 2 panels, and following resolution at day 30, shown in the right 2 panels. And what I want you guys to see here is that comparing the top and bottom square of each column, at the peak of inflammation, we see the skin get way thicker and more messed up than it normally looks. While after inflammation resolves, the skin looks normal again. So this resolving feature of the imiquimod model is really important since it specifically allows us to distinguish the short-term effects of the direct inflammatory response from long-term effects that stick around even after inflammation goes away, and that could be implicated in a lot of the chronic disorders that we've discussed. So having gone through this model, I can now show you guys the key experiment of the study that really jump-started our whole line of investigation. So what Sham and Shruti wanted to ask is if inflammation changes the long-term ability of skin to perform its usual key function of repairing itself in the face of environmental assaults. So to do this, and this is just following the top, they gave inflammation, let inflammation resolve, and then wounded mice and compared the rates of wound healing between inflammation experienced versus inexperienced skin. And the reason they used wound healing is that first, wounding is itself an inflammatory stimulus, so we can really see how an inflammatory response at one time impacts the response later. And second, wounding gives us a really nice quantifiable metric, specifically the rate of wound healing, to evaluate the extent of the inflammatory response. And while we're starting to play around with different secondary inflammatory stimuli besides wounding that more accurately model inflammatory diseases, the responses to these other stimuli aren't as clean to measure, especially for a first pass experiment like this. And really fascinatingly, when Sam and Trudy looked at the wound healing rates in inexperienced versus inflammation experienced mice, what they found is that inflammation experienced skin heals wounds much faster than its inexperienced counterpart. So on the left is just a visual for this, and on the right is the quantified rate of wound healing across multiple mice, which shows that experienced skin heals wounds over twice as fast as skin that never experienced inflammation. So these results really suggest that the skin retains some type of quote unquote memory of previous inflammatory responses that allow it to respond faster the next time it sees inflammation. And really excitingly, or one really exciting finding that Sam and Trudy made is that this inflammatory memory was established across every single primary stimulus they tested. So we just saw imicamode, which models psoriasis, and we see this same trend of results when we use a different inflammatory stimuli. Namely, they tested MC903, which models atopic dermatitis, as well as tumor-promoting agent wounding itself and fungi, and a species of fungi, all of which induce a very distinct set of inflammatory responses. So these results really suggest that the retention of memory is really a hallmark of the inflammatory response, at least in the skin, and that this finding is likely to be applicable to a really wide range of clinical problems that are caused by a wide range of inflammatory responses. And really just to touch on this last point, I think we can already see at this early point why inflammatory memory is so interesting from a clinical standpoint. Since if the skin is hyperresponsive even after just one inflammatory experience, this might help explain why certain patches of skin keep becoming inflamed again and again, and often with greater severity, as well as why inflammation never goes away in persistent conditions. We can imagine that the skin becomes locked into a hyperresponsive state and just snowballs away from there. And excitingly though, this means that we may be able to treat these disorders if we can develop an eraser of memory, which takes our skin away from this primed state during resolution and allows for future inflammation to proceed normally instead of becoming hyperinflamed or exaggerated like we see in disease. However, to make this kind of eraser, we first really need to address some basic questions. And first, we need to know how inflammatory memory works. So one of the earliest questions that we need to ask toward this end is what cells are responsible for actually storing this memory? Or a little bit more crudely put, where does the skin store its memories? So I mentioned before that most responses to environmental assaults require a really diverse network of cell types, both in the skin's epidermis and underlying dermis, to act in concert to orchestrate repair or healing. So really any one of these cells can play a role in enacting inflammatory memory, and we're still investigating the roles of various components of skin in this phenomenon. However, in both Sam and Trudy's work and in our subsequent studies, really just one prominent candidate has emerged, and that's this bottom layer of stem cells. And since these cells are about to become the star of our show, I'll refer to them by their full name from here on, which is epidermal stem cells and are abbreviated as EPDSCs. So I won't bore you guys with the data on why these are the primary candidates for where the skin stores inflammatory memory, though I'm more than happy to take questions on it later. However, I think for now it's sufficient to point out that these cells are first really essential drivers of inflammatory responses in skin, and second, which is a point I haven't made before, that among the cells in the skin, these epidermal stem cells are uniquely long-lived, making them especially prone to storing long-term memories. And these special features of epidermal stem cells led Sam and Trudy to realize that if we want to understand how inflammatory memory works, and consequently how we can manipulate it to treat disease, one very important question that we want to ask is, how does inflammatory experience influence the long-term state of epidermal stem cells? So before jumping into their results, as well as the clinical promise of their results, I want to briefly just talk about at a basic level what I mean when I say cell state, as well as some methods that we as biologists use to get a picture of cell state. So briefly, when I say cell state in this context, I'm really asking two questions. The first is, what is the cell doing now at this inflammation resolved or post-inflamed time point? And second, what is the cell poised to do upon future inflammation? Or in other words, is the cell poised to act any differently than it usually does the next time it sees some inflammatory stimulus? And to get at these questions, and this is a bit of our eating our vegetables when it comes to biology, one really powerful method that we can employ is to look at what we call chromatin biology. And again, this is a bit of eating our vegetables, but I promise that everything is going to tie together in the end, and I won't take too long with this kind of more heavy textbook biology stuff. So almost all of us are familiar with this classic double helix structure of DNA. However, if we actually look at the DNA in our cell and zoom out, we'll see that it doesn't lie in just one big stretch of double helix, but it's instead carefully packaged by being wrapped around a series of proteins. So the combination of DNA and the proteins that package it is called chromatin. And this is really one of the most powerful tools that we have to get an image of the cell state. So at the simplest but I think really applicable view, the chromatin in any given cell will look like a mix of these loosely packaged regions and these tightly packaged regions, which are shown on the left and right respectively. And I think we can just visually see that the DNA within these loose regions should be more accessible to the various machinery of our cells that interact with DNA. So we call them accessible, while the tight regions are less accessible to the machinery of our cells since they're so tightly packaged. So we call them inaccessible. And what this means for the cell is that it can control what DNA its machinery has access to. This determines what RNA gets produced from this DNA, and consequently, what proteins and downstream functions the cells can actually enact. In other words, the chromatin landscape of a cell is one of, if not the primary determining factors of which genes actually end up being used by which cells. And one of my favorite ways to really reflect this point is to remind everyone that almost every cell in our body, say a heart cell and an eye cell, will share effectively the exact same genetic code, even though each cell does something very, very different. And really the main thing that allows for this diversity of function from the same genetic code is the chromatin landscape, which again defines which genes are used by which cells. And really over the last 5 years, we've had an explosion in terms of technology that allows us to probe the chromatin landscape and get a sense of cell state. So the main technology that we use for this is a method called ATAC-seq. And I've written out its full name just for anyone interested in learning more about how this technique works. And I'll be happy to go through that in questions as well. But I think the really important point to get here is that ATAC-seq gives us a readout in which we can see how accessible chromatin is at any given point in our genome. So just as an example, I've shown some of our data for normal epidermal stem cells, and we can see in chromatin that we know should be accessible on the left, since the associated gene is highly transcribed, that our ATAC-seq data shows a high signal, right? We can see just a lot of black bars there. Conversely, in chromatin that we know shouldn't be accessible, and on the right, I'm showing a gene that's canonically not expressed in the skin, it's actually expressed in eyes, that the ATAC-seq signal shows little to no readout. So having established the importance of chromatin accessibility and understanding the cell state, as well as some of the tools that we use to probe the chromatin landscape, we can now really get at our question of how inflammatory memory actually works by specifically asking, does inflammatory experience change the long-term chromatin landscape of epidermal stem cells? And I'll jump the gun here. I'll jump the gun a little bit here just to orient everyone further as to why we're asking this question and say that if we do see these changes in the chromatin landscape, that implies that there could be long-term changes to cell state, which brings us back to our first two questions of one, is there a change to which genes are actually used by these cells? And second, is there a change to which genes are poised to become used upon future inflammation? And given these questions, we'll just first look at the broad chromatin landscape. So to do this, Sam and Shruti performed ATAC-seq on epidermal stem cells taken from mice that were either inflamed, resolved through day 30, or resolved 5 months later through day 180, just to see if any effects that we think stick around really do stick around for a long time. And excitingly, we see that this is actually the case. So I'll just walk you guys through the figure here. And briefly, the gray kind of peaks are from untreated epidermal cells. The red ones are from treated epidermal stem cells, and the black bars on top of each peak shows what our computational programs call out as a region of accessible chromatin. So for each gene, which is shown on the bottom, we read the figure top down, and I'll just follow with AIM2, which is shown on the very right. since I think this is the cleanest example of what I want to show, but you can do the same with any of the genes in this figure. So what we can see, just following AIM2 on the very right, is just visually at the peak of inflammation, or at day 6, 2 new chromatin regions become accessible that weren't accessible before. And interestingly, these regions stick around even after inflammation is resolved at day 30. And fascinatingly, at least this left region sticks around even 5 months later. And we can see that even this right region has some accessibility, even though our computational program isn't calling it as an accessible region. But really overall, what this tells us is that epidermal stem cells can remember inflammation, can remember inflammation by changing their long-term chromatin landscapes, and that at least some of these changes can last a very long time, even almost 6 months after inflammation goes away. And just to give you guys a sense of what this looks like across all chromatin regions, and not just the ones I've shown as examples, we can also compare the accessible chromatin, or all accessible chromatin in post-inflamed, or PI, versus untreated or control epidermal stem cells, and ask how similar these landscapes are. And interestingly, we can see that about 10% of these chromatin regions within post-inflamed epidermal stem cells are unique to these cells, just like the chromatin that's being shown on the examples in the left. And we call this quote unquote memory chromatin. Okay. So really from here, we can start to ask our 2 follow-up questions, which is, one, given these chromatin changes, does inflammatory experience change the genes that are actually used by epidermal stem cells? And second, does this poise any genes to become used upon future stimulation? So one relatively easy and widely used way to look at what genes are actually used is to collect all the RNA in a cell and see how much RNA from each gene there is, using a technique that we call RNAseq. So our experimental setup here is similar to what we had before, where we essentially just take epidermal stem cells from post-inflamed mice, and then this time profile their RNA instead of their chromatin, which allows us to understand if inflammatory experiences change not just the chromatin landscape, but also which genes are used that arise from that chromatin landscape. And the experiment to get at our second question of whether any genes are poised to become used upon future inflammation is a little bit more involved, but I still think quite elegant. And for this, Sam and Trudy wounded mice, and then looked immediately after wounding to see if any genes are expressed significantly more quickly than they are during the usual inflammatory response to wounding. And I'll end this data part of my presentation by showing you guys that it's really the second option that seems to be at play. So we can see this here, and just walking through the figures, each dot in this figure is one gene, and its position on the x-axis is its relative level of presence or expression within the cell, and its position on the y-axis is how much more or less expressed it is in inflamed or inflammation-experienced stem cells versus the untreated or inexperienced counterparts. And if a dot is red, that just means its up or down regulation is statistically significant. And for anyone who cares about the statistics, they used a false discovery rate, or FDR, threshold of 0.05 to control the rate of false positives that were called from this data set. And what we can see is that during inflammation, really like we'd expect, there are these really major shifts to what genes are transcribed because these cells are really stressed out and inflamed. But that once inflammation resolves, in this middle graph right here, transcription, which again, we're just using as a measure of overall gene use, largely returns to normal, save for a very small handful of genes. However, immediately after wounding, we see that a number of genes, specifically 268 genes, show rapid regulation of transcription. And remember that in this rightmost plot, both inflammation experienced and inexperienced mice were wounded. So what a red dot means in this right plot means that the associated gene is regulated at a significantly faster rate than it usually is during the wound response. So this last point is exciting, and it suggests that a key way that inflammatory memory may work is by remodeling chromatin to prime the rapid use of certain genes. And for this, we focus on this subset of 140 upregulated genes, which we refer to as quote unquote poised. However, we really want to connect the pieces of our puzzle and make sure that these 140 poised genes actually have something to do with inflammation. And second, that these genes are actually associated with the memory chromatin we saw earlier. And in this regard, we see that both of these are true. So on the left, we can see that over 90 or almost 90% of rapidly upregulated or poised genes were genes that were also involved in the normal inflammatory response at day 6, suggesting a very strong association between these genes and inflammatory responses. And second, we can see that over half of these genes are associated with memory chromatin. So half may not seem like a lot, but just for reference, I'd like to remind you all that the odds of any one random gene in our genome being associated with memory chromatin is vanishingly small. So this 50% number really suggests that chromatin remodeling plays an important role in the poising of inflammatory response genes. In addition, we've since done some more complex but currently unpublished work that's pushed this 50% number closer to 80%. So we really do have a good sense that chromatin changes are closely associated with the poising of genes within inflammation-experienced epidermal stem cells. And now we can finally put our picture together to have a model by which inflammatory memory works, and from which we can build on to develop new treatments for associated diseases. So just to summarize, we know that inflammation results in a lot of chromatin changes, and that a subset of these changes are maintained in the long run. We further know that, at least with the psoriasis model that we studied here, or that Sam and Trudy studied here, that these changes don't lead to major changes in gene use by the epidermal stem cells during normal conditions. However, these stem cells are now trained so that the next time they see inflammation, they rapidly upregulate inflammatory response genes and become more intensely inflamed. So this model is especially important since it gives us our first mechanistic insight into how inflammatory memory works. And it also tells us that if we want to look for an eraser of memory to treat associated disorders, that we should really look specifically within epidermal stem cells. However, as promising as these findings are, a number of really pertinent questions remain, and I'll focus on a couple that are especially relevant to our disease-oriented goals. So we first want to understand at a deeper level how inflammatory memory is established and maintained, both out of biological interest and with an eye for finding specific molecules that we can target to erase memory. So Sam and Chris Kelly are wrapping up a study showing how this works with our psoriasis model, or imiquimod, and Chris and I are digging into how this works with distinct types of inflammation. And second, it's also of interest to know whether inflammatory memory impacts more than just poising transcription, and this is a question Chris and I are working on as well. Lastly, more down the road, we'll want to apply these findings to human disease. So this point isn't a major area of focus for us right now, although Chris and I are doing some work toward this end. However, I think it's worth noting that on the heels of Sam and Trudy's study came a paper from the Shalit group out at Harvard, which showed that stem cells in our airway, or respiratory stem cells, maintain an inflammatory memory to allergic inflammation in human patients with chronic airway diseases, which locks in these stem cells into a hyperinflamed state. So this study really suggests that inflammatory memory is pertinent to humans, and that this relevance is very likely to extend across multiple organ systems, and consequently multiple types of disease that go beyond the skin disorders that were the focus of our presentation here. So before I leave you all for questions, I'd just like to quickly summarize what I've shown, and all under this kind of tagline in the title, that basic science is really critical for defining treatment paradigms, or the way we think about treatments for global health challenges. So I specifically discussed this concept in the context of inflammatory skin disorders, which represent one of the most prominent, and burdensome global health challenges out there. And I talked about how our lab's work looking at the basic biology of the skin's response to environmental stress revealed an inflammatory memory that is carried specifically within stem cells, and which opens up an entirely new and really promising treatment strategy for these currently uncurable set of diseases, which revolve around finding quote unquote erasers for inflammatory memory. However, big picture questions still remain, a subset of which I mentioned at the very end. So this really does remain a very exciting and active area of research. Okay. So with that, I will thank you guys all. And just before turning over for questions, I'll quickly plug my emails if you guys want to reach out to me with any questions. I know a lot of you guys must be in really pivotal stages of your lives right now, being in high school, and I'm really happy to address any questions you guys may have, like finding research opportunities or setting up research projects, applying to colleges, choosing different fields, some of my own work, and so on. And if you do want to reach out, try the Rockefeller email first, though I know sometimes that email sends things to spam kind of quickly. So if you don't hear back soon, then you can also try my Columbia email, which I kept from when I was an undergrad. So with that, I'll thank you guys all for your time and turn the floor over for any questions.
Thank you so much for the amazing presentation. That was really insightful. Thank you. So we will now begin the Q&A session. We'll first start with the questions that were asked through the registration form, and then we'll get to any questions that were asked in the Zoom chat. So this is your opportunity to ask any questions that you have. So ask whatever you have in your mind. I will now hand the floor over to one of our board members, Mark, to read the questions that were asked. Thank you.
Thanks, Adam. So as Adam said, we're going to just read off some of the questions that we were given in the Google form. So firstly, we have, what is the most ethically challenging scenario you have faced in your career?
Yeah, I think that's a really good question. So I think on one end, I'm kind of fortunate to be, well, on one end, I am still in a very early phase of my career, so I haven't faced too many direct human-related ethical challenges. I will say at this kind of introductory phase that I'm in right now, one really prominent challenge though is or as an experimentalist, is the way that we treat the animals that we work with. So I think one thing that a lot of people who want to get into biology or biological research should consider is that, yes, we're always doing our research with an eye toward having some benefit for human health or doing some good for the world out there. But that in order to do that, really the best essential models we have require us to do some kind of screwed up things to the animals that we work with. And I think something that's really important to learn is how do you set up projects and experiments that give you the results that you want while also treating these animals as humanely as possible. I think, yeah, so that would be the first one that comes to mind.
All right, thanks for that answer. So next question we have is, what tips can you give high school students who want to pursue a career in the medical field?
Yeah, so that might be a bit medically or too clinically oriented for me since I'm a basic biologist, but I can give some insights. So I think it depends on what kind of aspect of medicine you want to get into. If you want to get into practice, I know a lot of high schools have a lot of medically oriented clubs. My high school in Texas had, I forget what it stands for, but HOSA, H-O-S-A. And I know a lot of people who ended up going into med school or going into the practice side of medicine, got their kind of first experience of medicine through that organization. And also, if your school has any like clinical rotation programs, definitely explore those as well. If instead you mean more of the medical research side of things, I'll save that for a bit, since I think one question specifically asked, how do you get into research? How do you get research opportunities? So yeah, I can save that to when we get to that question.
All right, thanks for that answer. So the third question we have is, what challenges have you faced that made you who you are today?
Yeah, I think that's a really broad question, but it is a good one. I think though, I'm not so sure, or I think the way I like to look at it is that really kind of every experience that I've had has shaped who I am, and will continue to do so going forward. And one thing I don't like to do too much is try to force those experiences into one kind of narrative or anything like that. Actually, that's one kind of big gripe I have with the college application process and a lot of graduate application processes as well, is that they try to tell you to, or try to make you really condense your whole life into one clean story, when I don't really think that's the case at all. So I think, yeah, certainly I've gone through a number of challenges, both, spending long hours in lab, going through like mental health stuff, like et cetera, et cetera. But I think really each of those has shaped me in their own way. And it's kind of hard to pin an articulate answer down into exactly how those shaped me. So yeah, I'm sorry, I can't really give a great answer there. I think the best thing I can say for me is that the only thing I've really tried to put my effort on is on having fun with whatever I'm doing. And as long as that's the case, I feel like I haven't had to worry about, or haven't had to think too much about, how specific challenges or specific events have made me what I am now.
All right, that was well said. The next question we have is, are there current studies that show stem cells can be used for tasks such as growing a new heart cell to combat chronic heart disease?
Yeah. So this is also a little bit outside my field, but this question of but yes, there are, and I'm not sure exactly how far those studies are. But really, one prominent use of stem cells, if not really the most publicly acknowledged one, is using stem cells for regenerative medicine, right? So you have some organ of yours that's messed up, for example, a bad heart, and using stem cells to make a new heart, for example. So yeah, I'm not sure exactly how far along those studies are in heart. I do know with skin that there have been multiple examples where patients with severe burns whose skin is irreparably messed up can receive a graft of new skin in which stem cells will form new skin for those patients and they can become normal again. So this is, yeah, sorry, I couldn't really answer your question with regards to heart regeneration, but yes, that is a very, I think, exciting and prominent field in general with stem cell biology, is how it can be used for regenerative medicine.
Yeah, thank you about the skin comment, even though there's no really clues about the heart yet. So the next question we have is, what is your advice to high school students who want to start becoming more involved with research?
Yeah. Yeah. So I think two things. The first is if there is some type of, I think, organized project-based science environment you can find in your school, absolutely, I'd recommend joining that. So when I was in high school, I joined science fair, I think either in 9th or 10th grade, and then continued it all through the end of my high school. And I think really being pushed to think about my own projects and set up my own experiments and really go through the pipeline of a scientific project got me really, really excited and interested in science, and was a really critical kind of first step toward me being where I am now, and I'm sure toward wherever I'll end up in the future. And the second thing is reaching out, just reach out to professors and faculty. So there's, I think, two ways you can go about getting into a lab. The more common way is to reach out to professors and see if they're taking students. So this can either be through some formal program that a university might hold for high school students, or even if there is no formal program, you can just cold email professors that you find interesting and ask if they have any spots open in their lab. And the second thing, which is that you can try, which is closer to what I did, is if there's a project that you really want to try it for yourself, you can also just try cold emailing professors and to see if they have a spot open so you can train and actually perform that work in their lab. The success rate on that is much lower. I remember I reached out to I think almost 30 people and just heard back from one. But that one person ended up being a very, very close mentor for almost 4 or 5 years. So I think that can also be a very worthwhile strategy as well. It just really depends on what you want to do. So hopefully that was clear.
Yeah, it was super clear. Thanks for the insightful thing about the mentorship with professors reaching out. Next question we have is, have you ever felt uneasy seeing the clinical conditions some patients come in with, and how do you combat this feeling?
I do like that question. So actually just one quick point on the last question. If you guys do want help reaching out to professors, please feel free to email me as well. I've helped a lot of high school students, especially kind of get their first foot in the door with research, and cold emailing is really one of the main ways to do that. So I'm happy to kind of give you guys the templates that I've used and that I know have worked from when I was a high school student. Okay, so for yeah, do I get kind of uneasy with clinical presentations of these diseases? I think, so I actually have eczema. Right now my flare-ups are fine, but this summer they were especially bad. So I think, or eczema is also atopic dermatitis. So I think that overall I'm fine with those clinical symptoms. I don't get too uneasy about them, but there are, I think, but yeah, I will say, if you want to get into either basic biological research or some type of clinical practice, yes, if you are grossed out by those things, I think that is something that you'll have to get over. I think the good thing is that most people do actually end up getting over them with practice. I think it's completely normal to be grossed out by these symptoms. They are gross, right? There's no real way around it. But I do think the more you see them, the more you're experienced with them, and the more you actually work with them, the more you'll get used to it. So it kind of sucks, but really just spending time and and doing the work, I think will get you over those symptoms before, or will get you over that kind of uneasiness before you know it. And honestly, even if it doesn't, I think there are plenty of other really exciting and equally essential and important things that you could be doing. So it's completely okay to say, I've tried this out. I really just cannot get over how pussy the skin is. So I'm just going to leave, right? I'm not going to do this. I'm going to do something else.
Okay. So that was an insightful answer, honestly. And the last question we have from the form was, what is your advice to students who want to enter the medical field but are having a tough time deciding where to start?
Yeah, so I think I'd echo the answers I gave before. Really, if there's any clubs at your school or organizations that you can join, do that. And I know in my high school, they offered this clinical rotation program where students could rotate around the clinic at a local hospital and get exposed to different parts of medicine there. When it comes to medical research and biology, I think one nice way to expose yourself is just to go to a local university's page or a university that you can see yourself working in and just read about what the professors are working on. All of these pages will have just a full list of, or pretty much every page should have a good description of what the professor's working on, maybe some papers that they've released and so on. Yeah, and a lot of papers may be still inaccessible. I know when I was in high school, that was a huge problem, that I couldn't actually read a lot of papers because they were behind a paywall. There is a really strong push in science right now to make sure that these articles are actually accessible to the public and immediately so. So hopefully that gets resolved for you guys too. And there are also some really excellent journals that are purely open access, meaning that you guys can access them and read about that research, medically oriented or otherwise, even without having some type of access through an institution.
Okay, thanks for that answer. Now we're going to go to the questions that were asked in the chat. So the first one we have is, wouldn't implementing the eraser slow down the curation of the normally inflamed skin? And after treating that, would the skin redevelop memory?
Yeah, that's a really, really good question. So the idea with the eraser is that you wouldn't use it unless the person is diseased, when they're already in this kind of hyperinflamed state that just keeps getting more and more hyperinflamed the more and more they age or the more time passes. So the idea is that the eraser wouldn't impact how the skin heals itself normally, but rather what it's poised to do the next time it sees inflammation, right? So your skin would still resolve normally, but the idea is that when you give this eraser during this resolution phase, that now you're taking your skin from this trained state back into an untrained state, so that the next time it sees inflammation, these symptoms don't get exacerbated. And after treating, would the skin redevelop memory? I think that's also a great question. We don't know, but I would suspect the answer is yes. The skin will redevelop memory if we're not constantly giving it some type of eraser. However, I think for the case of, so when it comes to normal cases, this usually isn't a big problem. Our skin can heal itself fine, and this memory really doesn't present with any clinical manifestations. However, in the cases of like psoriasis or atopic dermatitis or chronic wounding, where we can see inflammation is already exacerbated, so this memory will just exacerbate it further and further and further. This eraser, if we do apply it, I think applying regular treatments of this eraser would be a very worthwhile treatment strategy that would be worth exploring. At least from my personal experience with eczema, I'm applying creams to my face like every day. So it's not like doing, regular or daily treatments is something that's too, I think, out of the blue when it comes to these kind of treatments, at least as far as I'm aware.
All right, that was an interesting answer. So the next question we have is, what keeps you motivated in the work that you do?
Yeah, so I think on, so I think at a really kind of basic level, I've tried a lot of different fields and a lot of different things out, like consulting, like finance, and like medical career itself, like practice. And these are all things I've very strongly considered, but I think having had some exposure to all of these, I think the place where I've really found myself the happiest and most excited and most stimulated was when I'm working in this kind of basic setting and within basic biological research. Obviously, that's not to say that it's the best thing out there to do. I think it's just innately the thing that I enjoy the most, and that's probably it. And I just enjoy it enough that it never really feels like work when I do it.
I like that comment about, just enjoying the work that you do and not really just like, forcing yourself to do something. It's important that we do that. Third question we have from the Zoom chat is, why would humans evolve so that previously inflamed skin would be hyperinflamed? Are there any benefits to this?
Oh, yeah, so these are actually really, really good questions. Two questions that our lab are very interested in. And yeah, so this question of why would humans evolve, so first to the question of why would humans evolve so that previously inflamed skin will hyperinflame. The idea is that there are both adaptive or good and maladaptive or bad effects to inflammatory memory. Given that this evolved, what we would expect is that the good effects have outweighed the bad ones. And as an example for one good effect, we can see this increased rate of wound healing, right? So we know that our skin is constantly exposed to things that can wound us. So we can imagine that, especially for someone, say like younger, that's going out a lot, that healing skin faster would be something that is beneficial, right? On the other end, I think some of these more maladaptive effects are really kind of phenotypes or things that we see as we get older. So for example, like a lot of these diseases are more exacerbated as we get older or with age. In addition, one kind of more recent set of studies we've been doing is to show that this inflammatory memory can also predispose stem cells to form cancers, which is just another maladaptive effect. But even those cancers are something that in humans would really only come as we are older, right? And so the idea is that as long as we have enough good effects before we're the age to reproduce and form progeny, then that's fine from an evolutionary perspective. And whatever bad effects happen are evolutionarily speaking fine, as long as they happen after we're able to reproduce. Yeah, and I don't know if you spoke out his second question of are people genetically predisposed, but yeah, I can go to that as well. So he asked, are people genetically predisposed to get inflammatory skin diseases, or is it caused by someone's environment? And the answer to that is both. There are genetic predispositions to inflammatory disorders. And like, for example, I think one very prominent one is if your skin cells already have some mutation that messes up their strength or integrity, then we're already kind of prone to these inflammatory diseases. However, often there's also an additional hit you need from your environment. So for example, with atopic dermatitis, one kind of prominent combination is that there's a mutation in a gene for the strength of your skin itself. But then there's also some allergen in the environment that induces an inflammatory response that really makes this, and it's the combination of those two kind of features that make this inflammation really exacerbated.
I liked how you said about both the environmental and genetic factors can kind of intertwine and kind of they need to work together to kind of bring a sort of response together. Mm-hmm. And unfortunately, since we're cut for time, this will be our last question from the Zoom chat. And it says, throughout time, does the skin lose some inflammatory memory, or is it a constant thing?
Yeah, also a really good question, and something that I'm actually investigating right now. So we'd expect that the skin will lose some of its inflammatory memory if we wait really, really long stretches of time. But the exact extent to which that occurs and the extent to which that occurs with different types of inflammation, I think are still pretty unclear to us right now. So while we do know that skin keeps inflammatory memory for a substantial period of time, we don't know exactly how long that is. We have some idea that it could be very long, just given that day 180 data that I showed you guys before. But again, that's something that we're going to have to test out. That is a great question.
Well, thank you so much for all those amazing answers. We're unfortunately unable to cover the rest of the questions, but feel free to reach out to Sairaj Sajjath if you have any pressing questions. And we would like to once again thank Researcher Sairaj Sajjath for taking his time today and to give us the introduction on some of the most advanced research in stem cell biology. Thank you all so much for attending today's presentation. And please use the link posted in chat right now by Adam to obtain your shadowing certificate. Thank you all so much for coming, and we look forward to seeing you in our next monthly meeting. And we'll stick around for a couple of minutes to make sure everyone has access to the link.